Can you hear me?
Yes, sir.
Okay.
Hello, sir. Shall we begin?
1 minute, I'm just opening.
Okay, sir.
The slides are visible. Hello.
Yeah, the slides are visible, sir.
Whether it is in presentation mode,
Yeah.
moving.
Yes.
Sir, shall we start?
Yes. Shall I start?
Yes, sir. A warm good afternoon to all. It is a great honor to welcome Dr. JAC FREDO, AR, Associate Professor at IIT BHU Varanasi, as the resource persons for today's session. Dr. JAC FREDO is an experienced researcher and teacher whose work covers many important areas such as biomedical signal and image processing, biomedical instrumentation, computational neuroscience, neuroinformatics, developmental psychology, machine learning, and drug delivery. He completed his PhD from Anna University and has done research at top institutions around the world, including San Diego State University's USA, NTU Singapore, and RWTH Aachen University, Germany. His research focuses on understanding the human brain and disorders like autism spectrum disorder using modern imaging tools and machine learning. We are happy and proud to have him with us today. We look forward to learning from his insights and experiences. Please join me in giving a warm welcome to Dr. Jack Fredo AR. Over to you, sir.
Thank you. Myself, Dr. Jack. I'm currently serving as an associate professor at the School of Biomedical Engineering in IIT BHU. And also, I'm leading the computational neuroscience and biology lab. And our lab mainly focus on analyzing autism spectrum disorder through brain imaging modalities like structural MRI.
Excuse me, sir.
Yeah.
Your voice is not audible.
Not audible. Can you hear?
Sir, I can hear you now.
Okay. So we are working on autism spectrum disorder for the last 15 years to identify the brain imaging markers, to identify the prognostic markers, and also early diagnosis. So like identifying the structural changes of the brain region, morphological changes of the brain region, and functional changes. So how delay in communication between one region to another region happens in autism spectrum disorder when compared to the typical developing or how overconnectivity happens between the brain regions in autism spectrum disorder when compared to the typical developing or normal participants. So here are few topics I would like to discuss on today, like nervous system, brain and spinal cord. So brain imaging techniques, I will be discussing only about MRI. I won't be discussing about the DTA and fMRI, and something like artificial intelligence, what is supervised machine learning, and how we can apply this machine learning or how we can integrate machine learning and neuroimaging for the diagnosis of ASD, especially how we can build a diagnostic model by using AI algorithms integrating MRI and ML, and also different kind of features or metrics we can actually measure from MRI, segmenting the brain regions and extracting morphological features, or without extracting the features or without segmentation, how we can feed these images into deep learning models and we can build an diagnostic model. So here is a nervous system. So we can broadly classify the nervous system in human body into central nervous system and peripheral nervous system. So the central nervous system comprises brain and the spinal cord. So you can actually see over here, so the color pink represents the central nervous system. So it composes brain and the spinal cord. And the remaining part of the body, so there it we call it as peripheral nervous system, or the remaining part of the body is controlled by the peripheral nervous system. So the brain and spinal cord are controlled by the central nervous system and the remaining part of the body by peripheral nervous system. So in the case of peripheral nervous system, we have a somatic nervous system and autonomous nervous system. So what is autonomous nervous system? So the involuntary activities like heart rate, excretion, respiratory. So they are not in our control, so they are called as actually autonomous nervous system. So other voluntary activities are called as a somatic nervous system. For example, eyes open and eyes close, hand rise and down, walking. These are all controlled by us, so that is a somatic nervous system. So in the case of autonomous nervous system, we have a sympathetic nervous system and a parasympathetic nervous system. So parasympathetic nervous system represents the normal activity of an organ. So heart beats normally, respiration happens normally, excretion happens normally, sweating happens normally. So it is an actual parasympathetic nervous system. So however, sometimes we are undergoing some abnormal activities in our body. So it is a sympathetic nervous system. Example, if we are running or walking, obviously, our heart rate may be actually increases. Obviously, our respiratory rate may be increases. Obviously, our sweat may be actually increases. Upon a part of time, the body reacts to it and take it to actually control to an actually parasympathetic nervous system. So this whole body, that is actually the central nervous system and peripheral nervous system are made up of nerve cell. So we can call it as actually a neuron. So where these neurons are, so we are more interested in brain. So we have central and peripheral nervous system. However, we are more interested on brain because we are worried about the neurodevelopmental disorders or neurological disorders that happens to subjects or human beings. So we are interested to know about the brain. So here we have brain and spinal cord in the central nervous system. However, we are only worried about how a brain is actually working on. So we can see here that the human brain is protected by the bone or the skull. So in between the bone and the brain, we have different layers. There are arachnoid pia layers and also cerebral spinal fluid will be there. And then we have the brain. So human brain, we have different regions in the human brain, like cerebral regions, cortical regions. So different ways we can classify these brain regions. Either maybe like lobes based upon the lobes or based upon the regions we can actually classify. Like here we have said like this whole region we are saying the cerebral cortex or cerebral region. And then the remaining regions, we are saying it as subcortical regions. So one of the example, basal ganglia. So it is in subcortical region, so it helps in movement and reward processing. Another one is subcortical region, thalamus. It is for sensory gateway. Another one is hippocampus, memory. And this cerebellum for motor coordination. Amygdala, responsible for our human emotion processing. Hypothalamus for regulatory body function. Also, we can also classify this brain, or we can actually study the different brain regions based upon the lobes. We can say that this is the frontal lobe. It is for planning, organization, and self-control. A temporal lobe for processing auditory signals, or an parietal lobe for temperature, taste, and touch, and an occipital lobe for sensing and perceiving mainly visual signals. So if it is a normal subject or a normal human being, so all these regions will be doing its own function without any problems. So however, if there is any problem happens, that may leads to like morphological changes in the brain regions. That may be actually the structure of the brain may be actually varies, and also the function of the brain also varies. So how we can study this structure and function based upon studying the neurons. So because that is a functional unit of the human brain. So here we can see the neuronal structure. So we have the dendrites on the one side that helps to receive the signal from other neurons to this primary neuron, where maybe actually under consideration. And then it consists of a nucleus central, and this information or the signals received through the dendrites will be passed through an axon, and then it actually receives an axon terminal. If it reaches a minimum action potential, then the neurotransmitters will come outside that decides whether signal should be passed to one neuron to another neuron based upon the ion channels. So how much actually sodium or actually potassium or chlorine ions are actually inside and outside, that actually based upon the ion channel opening or closing. So that is decided by our neurotransmitter. So like inhibition or exhibition actually happens based upon the signal transfer from one neuron to another neuron. And here you have this axon. This axon is covered by Schwann cell or actually myelin sheath. It actually acts as an isolation in order to reduce the loss of this transfer of signal from cell body to this actually axon terminal. And also we have the cell membrane and also the whole cell body we have. So now we know that the brain is have two colors, gray matter and white matter. So why it is gray and why it is white? So this region of the neuron, so this axon, it is covered by myelin sheath, or actually made up of Schwann cell. So this actually appears like an white color. So what the inner region of the brain is white, and this whole like cell body, so it looks like a gray color. So what the outer surface of the brain consists of the gray color, and it will be ulta in the case of a spinal cord. So inside it will be gray color and outside will be white color. So but we are more interested to know about the brain. So if you want to know about the function of the brain in normal or abnormal condition, we need to record the activity of the brain. So how we can record the activity of the brain? So we have different modalities like electroencephalography, functional near-infrared spectroscopy. So they are very simple modalities that can be used to record the activity of the brain. So however, these modalities can take the information from the surface of the brain only, until and unless you are not implanting the electrodes inside the brain. So these electrodes are kept on the surface of the skull. Maybe in the case of near-infrared spectroscopy, the light can penetrate maybe 1.5 mm or actually 2 centimeter. Otherwise, you can't actually get that deep information from the brain. So whatever the activity or the electrical activity happens on the surface of the brain or the skull, you can actually record through EEG. So this signal may not be sufficient to know the function or structure of the brain. So we need to sometimes image the brain and see what actually happens in the brain and its different brain regions. So one of the method that can be used for imaging the brain is magnetic resonance imaging. There are other modalities also like PET and SPECT images. Maybe you need to apply some dye and all. It may be partially invasive. Like in the case of magnetic resonance imaging, it doesn't have radiation much and it can give the soft tissues very legibly and with high resolution, like spatial resolution is high. So in the case of magnetic resonance imaging, so there are modalities like structural magnetic resonance imaging, diffusion tensor imaging or diffusion weighted imaging, and functional magnetic resonance imaging. So in the case of structural magnetic resonance imaging, you can actually get the structural or morphological changes. So in the case of DTA, you can actually see the fibers or the water molecule movement in terms of magnitude and direction. Also in the case of functional MRI, you can actually get the functional activity of the human brain with the images. So we are more interested to know about magnetic resonance imaging, how the magnetic resonance imaging data is recorded from the human body. So human body consists of different components. It consists of fat, it consists of sugar, it consists of muscles, and also it consists of water. So we are more interested to water because it has actually two hydrogen atoms and an oxygen atom. Here we are more interested to know about the hydrogen atoms. So it is like a proton with the positive and negative poles. So we can say that it is actually having a south and actually north pole. So it is a positively charged proton. So we are interested about this hydrogen atom. So at a normal condition in a subject, these hydrogen atoms are there, or we call it as protons. And their orientation will be in different direction. One will be this, one will be this, and one will be in this direction, and one will be in this direction. And they will be in different directions with a different precision frequency. That means they will be rotating its own axis in different frequency. So this is at the normal condition. Let's assume that this subject consists of water and he has the hydrogen atoms or the protons. So now we are keeping this subject in the magnet. So magnets, it is a giant magnet with a north pole, and other side it is actually a south pole. And obviously there will be a magnetic field will be developed. So we call it as B naught. And towards the direction of this subject, this B naught will be acting. And now what happens is all these hydrogen atoms, they were randomly oriented and rotating in different frequencies. They will align in a single direction. Either maybe it is parallel or anti-parallel to this magnetic field. So we had protons. So initially they will be rotating in different frequencies and they are oriented to different directions. So when you are applying magnetic field north pole and south pole, so these all protons will be aligned to in a direction to B naught, and they will be actually parallel or anti-parallel. So now the net magnetic field or net magnetization, we call it as actually longitudinal magnetization, that is parallel perpendicular to this subject. We assume it is in the longitudinal magnetization. So the total magnetization now, it is because of this magnetic field and these protons are aligned to parallel or anti-parallel. So the net magnetization is longitudinal. So this is the first phase of the MRI principle. So now we are going to the next phase. So what happens already I have actually explained. So when it is in under magnetic field, So all these protons are either parallel or anti-parallel to the magnetic field. So that is over here. And I said they are all will be in same frequency, they will be spinning. So that frequency we are called it as precision frequency or nuclear spin. And the frequency of this spinning depends upon two factors. One is the material, that may be human or rat or whatever may be, and also the magnetic field. So how much amount of magnetic field we are applying through this giant magnet now, which is the North Pole and South Pole magnet, how much we are applying. So the precision frequency is a factor of these two parameters. So the material we are using and also this actually strength of the magnetic field. So now we are applying a radio frequency pulse. So now what happens is the protons are in the parallel, they will be knocked down to another direction. So the parallel, maybe they are in parallel or anti-parallel. So once we apply this radio frequency pulse, they will be knocked down to another direction. And the net magnetization, we are calling it as transverse magnetization, that is maybe perpendicular to the longitudinal magnetization. And how much angle they are knocked down, that depends upon the strength of the radio frequency pulse and how much duration we are applying this radio frequency pulse. The two factors are here. So they are knocked down to another direction, and how much angle they are knocked down, that depends upon the strength and the frequency of the radio frequency pulse. And because they are knocked down to another direction, so the net magnetization is transverse and it may be perpendicular to the longitudinal magnetization. So longitudinal to the magnetic field. So we have the protons initially, they will be in a random positions, random frequency and random orientation. Once we kept them in the magnetic field, they are either parallel or anti-parallel. There will be a net magnetization that is in longitudinal direction. And now we are applying radio frequency pulse. So from the longitudinal magnetization, so it comes to transverse magnetization because these protons are knocked down to another angle. So this is the whole story we are summarizing here. So initially the protons are oriented in different direction and spinning in different frequency. So we are keeping them in magnetic field, and they will be either parallel or anti-parallel to the magnetic field, and net magnetization is longitudinal. We are applying radio frequency pulse, so they will be knocked down to another direction. That leads to transverse magnetization. So that is the whole story we are summarizing here. So once this net magnetization, once this radio frequency pulses, we are switching off. So again, the transverse magnetization go back to longitudinal magnetization. And this, because this knockdown protons will actually go back to its original position, so that the transverse magnetization go to the longitudinal magnetization. So while during this process, it may actually creates an electrical signal. So this whole process, like from longitudinal magnetization to transverse, from transverse to longitudinal magnetization, it actually looks like a spiraling based upon the free induction decay. So there will be an spiraling creates an electrical signal called the free induction. So we can record this electrical signal, and the amount of electrical signal released by the different regions may be actually varying. Based upon that, you will be getting different amount of current, and you can actually map it and you can actually create an image. So there are two more parameters are there. One is a T1 time and another one is a T2 time. So if you are recording the data, when 63% of the longitudinal magnetization is returned, so then it is called as T1 time and or T1 weighted. And the T2 time is like when it is actually decays 63% of the transverse magnetization. So that is actually in T2 time. And as I explained, so different parts of the body, maybe heart, maybe fat or muscle, they'll be having different amount of recovery time, T1 and T2. So however, there will be few limitations will be there. So this, because of this radio frequency pulse, only 90-degree pulses you can actually apply in the case of RF. And the signal may be decays rapidly. It is a small amount of current. It may actually decay very rapidly. And these protons, all the protons may not be getting equal amount of magnetic field. So when they are kept under this primary magnet, they all may not be having actually under same magnetic field. So those who are near to the magnetic field, they will be actually getting more, or actually away from the magnet, they will be getting actually very less. So the recovery period or the T2 constant, T1 or T2 constant maybe differs with respect to the body regions. That may leads to loss of the signal because we may not be able to actually get the current from the different parts of the body, at least current. So we will be end up with an holes in the images. That is no signal we are receiving. So we won't be able to map and reconstruct into an image. So that leads to an holes in the image. So again, I'm saying the story. So like protons are there, and they will be under primary magnetic field. There will be long-term magnetization, and then RF is applied. It will be transverse magnetization. RF is switched off, and then it will go back to long-term magnetization. There is a spiraling happens because of this. A current will be generated and will be mapped to make an image. So we said here. The different regions away from the maybe protons away from the B naught may not be able to release the current same as that, so it may release slowly. So in order to capture this, in between two 90-degree pulse, we are continuously applying this 180-degree pulse, so that even though it is releasing this energy very slowly, you can capture these signals. So this is the whole story about the magnetic resonance imaging. So how we acquire the MRI data, and by using an MRI data, you can visualize the soft tissues, you can visible the white matter, gray matter, you can see these lateral ventricles, brain regions, subcortical regions, the corpus callosum, brainstem, cerebellum, thalamus, hippocampus, whatever the brain regions, you can actually visualize, and if there is some morphological or structural variations, you can actually identify. So another part is machine learning. So we know machine learning. We can classify into supervised, unsupervised, and reinforcement learning. So supervised learning requires class labels. So it can be used for classification and regression. So if you want to classify the subject or any samples, maybe apples and oranges. And if you have your class labels, you can extract some features from the apple and orange. Maybe its color and shape. And you can actually give these features to the classification model, and it can actually build an model. And when you are giving a new image of apple or orange, it can actually say that whether it is apple or orange. So regressions, we are trying to predict. So if you have an data like last 3 years, the raining versus the weather condition, so we can actually predict like in the 4th year, so what it will be the climate, whether it will be raining or actually sunny. So in the case of classification, maybe cancer versus non-cancer. So if you have the images of cancer and non-cancer subject, and you can extract the patterns from the image, and you can classify cancer versus non-cancer. So in the case of biomedical application, again in the case of regression, like we are giving some drugs to the subject, cancer of subject, whether he is recovered from the disease or he is going worst condition. So if you have a time series data for 30 days, you can actually predict what will be the next day condition. So in the case of unsupervised learning, if we don't have a labels, class labels, we need to actually cluster the data. Let's assume that some disease condition, inside the disease condition, we don't have an annotation. Or subtypes or subgroups in a disease condition. We don't have a proper annotation criteria or a parameter for annotating the data. You can give to unsupervised learning, and you can actually cluster the data based upon some distance metrics, and you can actually group the data. So in the case of reinforcement learning, a continuous learning process happens from positive and negative outcomes. So if it is positive, it will be giving a reward, or if it is a wrong output, it will be giving a penalty. So it will be continuously learning based from the environment, like a chessboard. So based upon the correct, if it is a correct movement, it gives a positive reward. If it is a negative, bad movement, it will give a penalty. So continuously the model will be learning, and at a point it will become a best model. So unsupervised learning, we have discussed about clustering, but even if there is no annotation, and if you want to reduce the dimension of the data, so in the earlier case, we have said only 2 features, maybe an color and size of the fruit. However, sometimes when we are extracting voxel-wise features from an image, you maybe end up with maybe 30,000 or 40,000 features. And all those features may not be required to build a model, or they are all not significantly contributing for classification. So you need not to use all the features. You can use only the significant features. Or sometimes 2 features may be related. If it is a regular shape, sphere, the size and maybe volume and maybe the diameter may be linearly correlated. So you need not to use 2 features. You can avoid the correlated features, and you can use 1 feature. So at that time, you can use dimensional reduction. By using supervised and unsupervised learning, we can use dimensional reduction. And another one is an association. Also, you can use by using clustering. So what is the relationship between one and another? Like if you are buying a drink, there is a high possibility to buy a snack. If you are buying a shoes, there is a high possibility to buy a socks. Or in the case of disease condition, comorbidities, if a person is having autism, then high possibility of having ADHD. And if he's having ADHD,
വൈകുന്നേരം തീരുമാനം പറയും മാനേജ്മെന്റിൽ സീറ്റ് എന്ത് ചെയ്യാൻ ഞാൻ പറഞ്ഞു എത്ര മാസം ഞാൻ പറഞ്ഞു 3-4 വെയിറ്റിംഗ് ലിസ്റ്റിലാണുള്ളത് അപ്പൊ തീരുമാനം വേഗം പറഞ്ഞോളൂ
can we mute their mic? Can we mute their mic?
ഇന്നും മഴ രാവിലെ പോകും ഇന്ന് രാവിലെ മഴയുണ്ടല്ലോ അവിടെ
Okay, let's continue. Like comorbidities in a disease condition, if there is autism is there is high possibility for ADHD, and if there is ADHD, there is high possibility for epilepsy. So such kind of comorbidities, if you want to predict, we can actually use association principles. So this is not an exact figure, but here we can actually say, like if we have an MRI image, so either we can segment the brain region and then you can extract the features and you can go for classification, or we can feed this image to deep learning algorithm and we can go for the classification or we can extract the patterns by using deep learning algorithms, and then we can use machine learning algorithms for the classification. So our whole objective is how to integrate this autism spectrum disorder, the neuroimaging and AI or the machine learning for the diagnostic classification of autism spectrum disorder, or to identify the prognosis in the disease condition, or to identify the subtypes or subgroups in a disease condition, or to identify the comorbidities in a disease condition, or if you have a large number of features from a 3D data like a structural MRI, it is a three-dimensional data, how to reduce this actual dimension of this data or features reduction by using AI algorithm. So this is our whole research, and with the exact problem statement with respect to territory, with respect to region, with respect to the disease condition. So we need to have a suitable paradigm to record the data and then to process this data. So obviously, MRI machine may not be available in every centers because it's very costly. In 1.5 Tesla itself, it will be costing 15 crores to 16 crore. And if you want to have a research, you need to go for a 3 Tesla machine. And this 3 Tesla machine may be costing around 25 crore. So it is not feasible to install such kind of machines in academic institutions. And another level of challenge is, even if you install this kind of machines, the running cost may be actually higher. So you need to have a very good consumable and contingency amount, and you need to recruit some expert to run this machine. And another level of challenge is you need to recruit the participants to handle this autism. I don't know how many of you came across autism kids, the children. So to recruit and to handle them and to clinically diagnose them, it is highly challenging. And once it is highly clinically diagnosed, to record the imaging data, it is another level of challenge because they are not in control. And putting them in a dark room like an MRI machine with giant magnets running with a high noise, so it is really a difficult job. So we need to integrate some way some MRI centers. So here this slide we are trying to discuss about why we want to work on autism spectrum disorder, and why we want to go for imaging modalities, and why we want to use these AI algorithms, so that we are going to discuss in this slide. So autism, we say it as autism spectrum disorder. We called by using this name. Why it is this name? Because it is of different disorders combined: autism, pervasive developmental disorder, Asperger syndrome. So all these combined, it is called as autism spectrum disorder. And the symptoms are like deficits in social communication and social interaction. So they're having restricted and repetitive behavior patterns like running in a circle, flapping hands continuously, and they may not be able to communicate well with others socially, verbally, and cognitive difficulties will be also there. So statistics say that 1 in 54 children are identified with ASD. Recent study says it is 1 in 36. It is a big number. And if you come across from 2000 to 2020, around 241 percentage increase from the baseline. So it is the number of occurrences very high. So in the earlier days, we used to hear very randomly about autism spectrum disorder. However, in many families now we are hearing, and maybe it is because of the diagnosis or awareness has been improved, or on the other side, we can say that really the prevalence and occurrence is actually higher. Mostly it is happening to male kids when compared to females. It is like 4 times if you higher in common in males when compared to females because it is a genetic factors over there. So how we are diagnosing autism spectrum disorder? So if you come across these symptoms, that is the behavioral symptoms, so you can diagnose, or in their family history, someone who has already diagnosed, we can actually quickly have a look into it. Or based upon the clinical scores, based upon the cognitive test, verbal questions, non-verbal questions, they have different scores like ADOS, ADA, SRS. Based upon this, you can diagnose. However, there are challenges in the clinical diagnosis. So sometimes an autism kids may be interpreted as a slow learner, or a slow learner may be interpreted as an autism kid. And the symptoms of autism and ADHD are much similar, so they may be actually misdiagnosed each other because these parameters or these symptoms are like very subjective. It varies with respect to subjects, and it maybe varies with respect to culture and religion and territory also, region also. So their behavior and these scores are much subjective, so there is high possibility of misdiagnosis is there. So we are trying to look into an automated process, how to diagnose. So obviously we need to think about the physiology, and we need to think about from the brain patterns whether we can identify the biomarkers. So studies have reported like structural alterations are there, that is overgrowth or undergrowth of cortical and subcortical regions, maybe cortical regions or the subcortical regions like corpus callosum, brainstem, cerebellum, or thalamus, hippocampus, and also the connectivity between the brain regions. So how a brain region communicate with each other for specific tasks. If someone is calling them JAC, so it should go to auditory cortex. So it go to diaphragm, then to auditory cortex, and then it should go to the hippocampus. It should check whether he is calling my name or random name. So if it is calling my name, so it goes to motor cortex, and it actually I will be turning back and see who is calling me. So this is because of the brain regions are communicating. That means one neuron is actually firing another neuron, and the communication happens. However, in the case of these kids, the communication may not be happening in a regular way. There will be a delay in communication. Or in some case of cognitive processing or some specific verbal communication, this may be very higher. They may be hyperactive also. So in order to identify these patterns, we need to image the brain. So there are different modalities: functional MRI, structural MRI, and diffusion tensor imaging. So to start with, we are having structural MRI. So our lab is working in functional MRI and DTA also. But to make it very simple, I will be explaining about how we are using structural MRI for diagnosis of ASD. And the study I am explaining is the very preliminary study. Not explaining about very complex study, explaining about very simple study. So this study, why we did it is like a few problems we faced. So even though we worked in different places, especially the postdoc place where I worked in San Diego. So they have the MRI machine, they have a very good supercomputing facility, very good workstations to process this data. Really it is very challenging to process the MRI image, and very good clinical team is there. So same in Germany and San Diego, Singapore. However, when you start a lab in India, you will be having maybe a seed grant of 10 lakh, and you need to establish a very good lab. Means it will take time. So computational facility, we are thinking like, so such kind of computational facilities may not be available in every territory of the country. So we have thought like, if we have a very simple computational facility, whether we can actually target a specific region of the brain which can act as a biomarker, and without using these standard toolboxes. So in order to process these MRI images, we have different toolboxes like FSL, FreeSurfer, MIPAV. And these toolboxes take a lot of times. Maybe to process one image, it takes 12 hours, even with a very good computational facility. So we thought like whether we can develop a simple algorithm and we can target one region and whether we can make we can extract features from this particular region and whether we can build a diagnostic model. So such kind of biomarker, even if a person has a simple laptop who is actually remotely there, they have an MRI image somewhere they acquired and they want to see that really AST characteristics or symptoms are there, they can actually utilize our model. So the whole objective is we need to skip the standard toolboxes. We need not to look into all the brain regions. We'll be targeting one brain region, and the whole system should be computationally faster. So these are the specific objectives we had with respect to Indian system we are targeting. So, but we want to compare this with 3D models also. So we are segmenting the images from 2D images and 3D images, and we will be targeting one particular region, that is corpus callosum, and we will be extracting these 2D features, geometrical and sonic moments, and also we will be computing volumetric features, and we are trying to say that which feature is significant, so whether the simple features is enough to discriminate ASD and normal subject, or we require a complex 3D information. Obviously, we will be using machine learning models for building the classification model. So here is a simple process pipeline. So we have taken the original structural MRI image. We have taken one slice, for mid-sagittal slice we have taken, and we have used a simple level set algorithm. So it's a custom built simple level set algorithm. So we will be giving the initial contours and we will be segmenting the corpus callosum out. So why have chosen corpus callosum? So there are a lot of studies that says that the width of the corpus callosum or the dimension of the corpus callosum is less in AST, or the white matter intensity is very less in the corpus callosum in AST when compared to typical developing. So what they were not able to communicate, like left hemisphere and right hemisphere communication is less. Also in the case of, we have taken the 3D image also. We use the computationally costly FreeSurfer algorithm or the toolbox, and we have segmented this corpus callosum region, and we built a 3D model over here, and we computed the volumetric features also from the corpus callosum. And then we used the feature ranking algorithm, and then we built So here is the process pipeline. So we have taken the 2D image, and then we have made the initial contour, custom made it initial contour, and it settles at the edges of the and then we have segmented out the corpus callosum region. And here is the mask, and here is the ground truth image. And so whenever we are using a segmentation algorithm, we need to assess the efficiency of the segmentation algorithm. So whether it has over segmentation or under segmentation or correct segmentation it has done. So in order to assess that, we need to compare the segmented output with the ground truth. So we had the ground truth images. So we are comparing our segmented output with the ground truth. So there are different techniques out there. to validate this, like simple matching, Rogers and Tenement, Ham and Sokol and Smith, Pearson, neuron. Different metrics are there to compare it. So here y-axis is the each metric value, and this is with respect to each subject, what is the value. So we have made a box plot. 0 means no matching, 1 means very high matching. We can see that most of the subject, the segmented corpus callosum is the matching value is higher than 0.96. That means the segmentation was very good. A few outliers are there, still it is higher than 0.82 or 0.8. So we can say that the segmentation is good. So this is the 3D segmentation. So we have taken the 3-dimensional images from sagittal view. This is the sagittal view. This is the transaxial view. Then this is the coronal view. So around 256 slices in coronal and transaxial, and in sagittal it was around 171 slices. We have skull stripped the images. We fed this data to FreeSurfer, that is a software for pre-processing and processing the MRI data. It's a well-established data. FSL and FreeSurfer are well-established data for neuroimaging. And then we segmented the data by using Atlas. So in the earlier case, we used the custom-built software to segment the data. In the case of here, we are using the standard Atlas in order to pre-process the data. So what is Atlas? So it is an image with the predefined areas. Maybe neuroscientists would have actually segmented the brain into different regions, 520 regions or 400 regions, 500 regions. And we will be mapping that Atlas with our image. So it gives the specific actually boundaries of the brain regions. So here we are only interested in the corpus callosum region. So the districts and Atlas, I think it segments the brain into around 150 regions in both hemispheres. So, but we are specifically interested in assessing the corpus callosum region. So what we have done is, so we have only considered the corpus callosum from this output. So we have an 2-dimensional corpus callosum image that is segmented by using our custom-built algorithm, and a 3-dimensional image from the FreeSurfer software. So from the 2D images, we are computing geometrical features and discerning moments, and from the 3D images, we are computing the volumetric features. So we computed a lot of features like mean, standard deviation. So we have computed bounding box, extent, the major axis length, convex area, lot of features. And we have taken the mean and standard deviation for ASD subject, around 50 ASD and 50 TD. We have actually considered over here and around 0.487. So this is one feature, and 0.48473 for the typical developing we are getting. So not much variation we are able to identify, and the feature is not statistically significant. Same in the case of field area divided by extent, major axis divided by convex area divided by extent, eccentricity divided by convex area. So no feature is significantly contributing for classification between, discriminate between ASD and TD. Or we also extracted the moment features from the images, that is segmented images, 2D images. So one of the moment, like N15, M15, PAOH, so it is statistically significant, that is less than 0.05. And rest of the features are not statistically significant. Similarly, from the 3D volumes, we computed the volumetric features. So from the different parts of the corpus callosum, like anterior, posterior, center, we computed the volume. Even from the total corpus callosum, we have actually computed the volume, but none of them are statistically significant. So we thought of plotting this feature, how it actually distribution is across ASD and TD. So the x-axis is like ASD and TD, and the y-axis is the feature value. That is this N15, M15 PAOH. That is the only significant feature. So you can see the clear discrimination between ASD and TD. So that says that this feature is able to discriminate between ASD and TD with a small overlap, and their mean values are also extremely different. So, but one feature is not enough to build a machine learning model, and a lot of features also we don't require to build a model in order to reduce the complexity. So we have taken one data set. So all these data are actually obtained from ABAID. So we are also one of the contributor for this ABAID data set. So initially we have taken this KK data set site. So we did a lot of analysis, like we have taken the 10 percentile of the features. What we did is we ranked all the features, and we taken the top 10 percentile of the all features, and we build a model. 50% of the top 50% of the volumetric features, 20% of the geometrical features, 10% of the Zernick moments, all the Zernick moment features, all the volumetric features, all the geometrical features, and all the features. Also, we built the model. So what we see is, like, when you use all the features, the model struggles because all the features are not significant, and it affected the efficiency of the model. So we end up with around 60%. However, we have considered only the most significant features, 10% of features. We got the highest accuracy of around 69%. So this shows the impact of using only significant features to the models. The model won't be confused, and you can actually at least expect 10% of actually accuracy and improvement. So here, sir, we can see that we have used the volumetric features. So when you use all the volumetric features, it is 63%. On the other hand, when you use only 50% of the volumetric features, it is 61%. So that says that, so when we are losing some features also, it actually reduces the accuracy. So optimal amount of actually significant features we need to actually consider. So when you are using 20% of the geometry features, it is 59. When you are using all these actual geometry features, it is 57. So when apart from this using all the features, when you consider this as learning moments, so we will be getting a significant actually performance around 65% because we know that here learning moments are performing well when compared to the geometric and the volumetric features. So this is by using KK dataset and one RF classifier. In the case of ABED, there are around 18 sites contributed. So initially we considered only one site data, and we did this analysis, and by using this feature, that is 10% of all features, and that feature set we got the highest accuracy. So we run with the bagging and the support vector machine also, but we are getting lower accuracy, and we run for all the sites. So in the case of random forest OHSU, we are getting highest accuracy of around 72.69%. And then each site we have actually run site-specific models with a 5-fold cross-validation. As we can see over here, 72% in OHSU we got by using random forest, and you can see that from 60% to 72%. So variation is actually higher with respect to each site. That shows that in order to build a generalized model, it is very difficult. That says that a region and territory, there is a high possibility of actually variations of patterns in the ASD brain. However, we can see some features that are common between the sites. So let's example centroid. It was the significant feature in the case of the data used from UCLA, OHSC, and USM. And this feature is actually common between SDSU, GU, and USM. At least out of 7, at least 3 sites we can actually end up with an actually significant feature. So that feature you can actually commonly use for any subject from any region or any territory. So here it shows the feature rankings. So you can actually see, like, most of the features are from the 2D images. So these are from learning moments, So this is the geometrical features. So this is the geometrical features. These are zonic moments. So this is the geometrical features. So none of them are from actually, even if you consider on the top 20 features, none of them are from volumetric features, because these volumetric features are not contributing much. So you can say that when compared to 3D images, even 2D image is actually enough for building a diagnostic model. So this is the discussion we are actually end up with. So level set segmentation could segment the corpus callosum region, and it gives high similarity between the segmented region and the ground truth, and zonic moments extracted from the 2D segmented images is contributing well, and when compared to other classifiers, random forest is performing well for classification. So the limitations of the study is like we have built a site-specific model. We didn't build an global model, and also we considered the subjects ranging from 6 to 18. We didn't consider a broad range. Still, 6 to 18 is a broad range, but the other groups we didn't consider, and we build an actually site-specific models. So we can also extend it to other regions like brainstem, cerebellum, hippocampus, thalamus, and we can build an actually global classification model. So by here I am winding up. This is the very simple study we have actually done, but you can see my website. So we have done a lot of other studies also. So we are basically funded from ICMR, so, and SCRB, so they are funding to acquire the data from NIMHANS and Sri Chitrathirunal. In the case of MRI, and Inish is also supporting us. So thanks to my students. So now we are very good group with around 12 PhD students. They are working in different domains. At least half of them are working in MRI and OTC. So hopefully we will, over 5 years, we will give some benchmark results in the diagnosis and prediction of ASIET. So hereby I'm winding up. Thank you. If you have any questions, you can discuss.
Thank you, sir, for your wonderful session. Dear participants, do you have any queries?
Sir, good afternoon.
Yes, Pa.
Sir, how do you take or how do you give the importance to features? Sometimes you take 50% of volumetric features and 10% of these features, like that you are saying, no? So how do we conclude that how much amount of features is necessary?
Yes. So in the case of volumetric, we have computed only around 6 features. Like the corpus callosum is subdivided into some 6 regions, and then I think 5 regions, and then we have computed like anterior, mid-anterior, and the total volume of the corpus callosum, central, posterior, mid-posterior. So we had around actually very limited number of features over there. So we have actually thought like 50% we will utilize and we will see. So however, in the case of learning moments, there were actually more number of features were there. I don't know exactly the number, maybe 50 or 60 maybe. So we thought like 10% we will utilize. And in the case of geometric, so in the case of geometric also, we had actually more number of features, maybe around 30, 40. So we thought like actually 20%. So this is one hypothesis we followed. However, we also did for actually other number of features also. But this feature, we got actually significant accuracy. So we considered like 2 factors over here based upon the number of features we had, because our objective is to build a light model, simple model. So based upon the number of features, we have actually 10% or 20%. But if you see some another study, this is also a very simple study. Like okay, in this study, we see like one more slide. Yeah, so around actually 2000 or 3000 features were there. So what we did is like 0.1% of top features, 0.5% of top features, 1%, 1.5%. Like this, we have actually done up to 100%. And whatever the feature set, we got the highest accuracy that we used for testing the model. So while we are training, we will be using different feature set for 0.1, 0.5. So different types of number of features we have used. We have built the model and tested the model. So likewise, also we will actually analyze the data. In the case of fMRI, so if you compute the functional connectivity, you will be end up with if you consider at least 200 regions or 230 regions, you will be end up with 30,000 regions. So then this, what we will do is we will actually take top 500 features, top 1000 features, top 2000 features, because if you take 0.1, 0.5, then you need to run a lot of time. So it is computationally very costly to run all the time. You need to optimize the parameters of the model. You need to do 5-fold or 10-fold cross-validation, and you need to optimize the number of features. And some models like tree-based models, it will take a lot of time. And if you go for deep learning models, it will again actually requires a lot of computational cost. So based upon the study, sometimes we need to compromise, and we need to actually choose the number of features. Does it make sense?
Yes, sir. Thank you so much. Nice explanation. Thank you, sir.
Any other questions?
Any more queries?
Shall we end up?
Thank you, sir. We truly appreciate the time, effort, and insights you have shared with us today. Thank you, sir. Thank you, participants.
Okay, but thank you.
Thank you all.